Hello, welcome to my Blitz presentation. I have no conflicts of interest to disclose, so let's get started. Chronic pain is a daily reality for 1 in 5 Canadians, or nearly 8 million people, who suffer from this condition in Canada. And this condition is associated with a host of negative quality of life outcomes. Current research shows that chronic pain is largely a result of changes in the brain as individuals transition from acute to chronic pain. While most research is focused on following people as they transition to chronic pain, it is important to understand how we can distinguish within a group of people between who has chronic pain and who is otherwise healthy. Identifying these neural abnormalities can be used for better classification and targeted therapeutics, but research in this area is lacking. One region in the brain, often called the primary reward center due to its role in hedonic processes, like consuming drugs, is slowly coming into the spotlight in chronic pain research. This structure is called the Nucleus Accumbens, highlighted in the green blob. Recent evidence points to the nucleus accumbens as a region that changes from acute to chronic pain, and that the connection between the nucleus accumbens and the prefrontal cortex is particularly important. While this research shows that the nucleus accumbens is important in the etiology of chronic back pain, no one has systematically mapped the connections that distinguish between healthy controls and chronic back pain patients, so my research is centered on finding these differential nucleus accumbens connections. In my research, I used resting state magnetic resonance imaging scanning, where participants' brains were scanned while they were at rest. I was interested in how the nucleus accumbens was connected to other brain regions, and how this was different in chronic back pain patients relative to healthy controls. In the initial data set, I had 39 chronic back pain patients that had 2 resting states taken, and 41 healthy controls that had only 1 scan taken. I further subdivided the nucleus accumbens into its 2 subregions, the shell and core, that are known to have differential anatomy and connectivity patterns. The nucleus accumbens shell and core were analyzed to determine which regions the nucleus accumbens was more connected to and regions where the nucleus accumbens was less connected to in chronic back pain patients relative to healthy controls. These significant connections were then grouped into different resting state networks and correlated with a measure of pain called the neuropathic pain scale in chronic back pain patients to see if these connections are associated with clinical measures. Finally, we assessed if these connections could be reproduced in a separate dataset, our validation dataset, in 30 healthy controls and 18 chronic back pain patients. After whole brain correction, connectivity contrast revealed differential connectivity patterns between chronic back pain and healthy controls. Panel A shows a sample of the significant connections where the nucleus accumbens was more connected to in chronic back pain relative to healthy controls, and Panel B shows the opposite. Overall, these results show that the regions in the different contrasts can be grouped into larger brain networks, and that there is hyperconnectivity in chronic back pain patients to executive and default mode network regions such as the prefrontal cortex, and hypoconnectivity to language, memory, and salience regions such as the insula and hippocampus. I next assessed which regions were also able to predict clinical outcomes in chronic pain population using the neuropathic pain scale. I correlated the core and the shell of the nucleus accumbens with significant chronic back pain and healthy controls contrasts, and I found 8 connections that were significantly correlated with pain scores, 4 of which are shown here. In the chronic back pain greater than healthy controls, these were the left shell and core with the right dorsolateral prefrontal cortex, and the left shell to the anterior division of the dorsal medial prefrontal cortex. These were the positive correlations, and our only negative correlation was in the chronic back pain less than healthy control contrast, which was the connection between the left shell to the right temporal occipital cortex. These results show that chronic back pain patients who have higher pain scores also tend to have higher nucleus accumbens to prefrontal cortex connectivity and lower nucleus accumbens to temporal occipital cortex activity. Finally, we looked at these 8 significant connections and plotted their connectivity values in healthy controls and chronic back pain patients in both the initial and validation dataset. Only 2 connections, the left shell to the right dIPFC and the left core to the right dIPFC, were reproduced in this analysis, whereas chronic back pain patients had greater connectivity values relative to healthy controls. These results narrow down which regions can reliably distinguish between healthy controls and chronic back pain and also predict clinical outcomes. Broadly, this supports nucleus accumbens hyperconnectivity to prefrontal cortex regions and hypoconnectivity to language, memory, and salience regions. Specifically, the nucleus accumbens connection with the right dorsolateral prefrontal cortex seems to be an important region that differentiates healthy controls from chronic back pain, and that this result is reproducible. Further research should look at even larger datasets to confirm our findings, and this research can pave the way for specific biomarkers in chronic pain and eventual targeted therapies that avoid addictive pain medications used currently to combat chronic pain. A huge shout out to my lab, and here are my references. Thank you so much.